Whereas in our previous videos, we studied that how we can synthesize or retrieve small molecules from different databases. In this video, we will discuss that how we can obtain macromolecules like protein, DNA or enzymes from different databases, because we cannot synthesize these proteins or DNA in their proper 3D folding. So thousands of researchers around the globe deposit their crystallographic data every year in different databases. So the most important and the common one is the protein data bank, RCSB protein data bank, which contains 1,73,537 biological macromolecules. We can deposit our crystallographic data in this database. Like recently we deposited our crystallographic data of human carbonic anhydrase 1. We can search or visualize in different 3D views or protein feature views, and we can analyze different parameters. So today we will discuss that how we can use it. So for example, let's suppose I know that the PDB ID. So we should know that PDB ID is the unique code for each macromolecule, which is given depending on its type and the source from where it is obtained, like 6Y00. We can also write a name of any enzyme or macromolecule, and we can search that. So I have written here that it is the crystal structure of human carbonic anhydrase 1 in complex with our benzene sulfonamide derivative. So it belongs to lyases. The organism is Homo sapien, and here is the deposited and release date. This is our data, and X-ray crystallographic resolution is 1.37 angstrom. Here we can observe its 3D structure in detail. Like I want to observe 3D structure. So here we have 3D structure of that, which contains these are the sequences, and here is the ligand. Okay, now we should know some important information about any structure that we want to retrieve. Here is the publication regarding this data and other tools. So it contains two chains. It is a dimer A and B with 261 amino acid residues. Organism is Homo sapien. There is no mutation, and the gene name is CA1, and this is the enzyme commission number of this carbonic anhydrase. And here we have a ID for that ligand which is bound with this target O5T. Like PDB ID for macromolecule, there is also ID for ligands. Here we can observe 2D structure of the ligand, and here we can observe 3D structure of the ligand and ligand interactions. So we can observe also here that how it interacts with different residues. Like let's suppose this is glutamine 93 from carbonic anhydrase 1, and this is other residues. So now how we can retrieve this structure? Here is the download file. We can obtain FASTA sequence of that protein, or we can obtain its PDB format in 3D structure. So now I'm going to download this structure, and here is our structure. This is the dimer of human carbonic anhydrase 1. Okay, and here it gives information that this is our chain A, this is our chain B, and these are the ligands which are attached. Zinc is the metal ion present in the active site, and here is the ligand. Similarly, B is also bound with ligand, and these are the water molecules present in this structure. So in our next videos we will study further that how we can how we can observe ligand target interaction, and how we can modify